It's a real delight to be able to come and speak and see that this topic has gotten such a large audience in the past few years. In fact, when I first started talking to immunologists about memory in innate cells, they told me that that couldn't possibly exist. But I said, you have to be open-minded about this and say, memory is you remember what happened to you previously. There can be two types of memory. One is obviously antigen-specific. This is what has been classically thought of for B and T cells. But I'll show you, in fact, NK cells can do antigen-specific memory. But you can also remember non-antigenic responses, many of which we've heard about here today in hematopoietic progenitor cells, in macrophages, in lower organisms like Drosophila and snails. So the story I'll tell you today started when we appreciated that NK cells really care a lot about cytomegalovirus, both in humans and in mice. Humans who have no NK cells have had problems dealing with herpes viruses. And some 20-odd years ago, Tony Scalzo showed that, in fact, NK cells in mice are needed to keep them alive. So fast forward, we now know how this works. When you have cytomegalovirus infection, you first get the activation of myeloid cells through toll-like receptors to produce a lot of cytokines like IL-15, IL-12, type 1 interferons. NK cells constitutively express receptors for those cytokines, and they get all stirred up. They get activated. But they need to know where to go and how to recognize the virally infected cell. Well, it turns out that CMV encodes a glycoprotein which is specifically recognized by an NK receptor known as LY49H. This allows the NK cells with this specific receptor to bind to, attack, kill, and destroy those CMV-infected targets. They can also then start secreting various cytokines, and they can proliferate. If you don't have this LY49 receptors, the NK cell still gets stirred up by the cytokines, but the mice die because these NK cells can't find the infected cells specifically and target them. So this receptor, which is driving this response, is a member of the LY49 family. It signals through an adapter protein that we identified a number of years ago that I nicknamed DAP12. I found it because it looks just like CD3. I was doing homology searches, and this has a canonical ITAM, the same as in CD3 and the CD79 molecules that signal through B-cell receptor. You get some co-stimulation through a second adapter protein we named DAP10. It has a motif, in fact, that looks like CD28 and binds to PI3 kinase and VUV1 to amplify that response. This receptor specifically sees a viral protein, gene number 157, that encodes a glycoprotein, which we crystallized this with Chris Garcia's lab. It looks like a class 1 molecule, and this allows the NK cell to dock to the infected cells, kill them, make cytokines, and proliferate. Well, a few years ago now, I had a new postdoc in the lab, Joe Sun, who trained with Mike Bevan and had worked on CD8 memory. And he asked me, what happens to those NK cells after they've first seen CMV? Do NK cells have memory? Well, I'd read the textbooks. The textbooks told me NK cells only live for about 10 days. So I said memory would be pretty irrelevant if that was true. But we kind of looked back at the literature and said, well, nobody's actually tracked the fate of an NK cell to see what happens to those cells which had encountered the virus. So we used a nice system in that we had mice that were knocked out with this LY49 receptor, otherwise completely normal. These mice are sensitive to CMV infection. But now you can adoptively transfer in NK cells that express the CMV receptor, use a genetic tag, and they're the only NK cells that will have this receptor in this animal, infect with CMV and ask what happens to them. You can count them and see how long they lived. So the surprise was when you put these cells in and track them, they clonally expanded like T cells. They could go up to 1,000-fold expansion in the liver and 100-fold in the spleen. They then started to decay, but the real surprise was when Joe came back 2 months later and says they're still there. They weren't supposed to be. So this suggested that, in fact, first, NK cells can be long-lived, or their progeny can be long-lived. And it begged the question then of, would these cells expressing this receptor behave better the second time they encountered the infection? Would they really have useful immunological memory? So I remembered an old experiment that Ray Welch did about 20 years ago. He observed that if you take newborn baby mice and infect them with CMV, it's lethal. Newborn humans also have trouble responding to CMV. Ray had shown a number of years ago that if you take adult NK cells and put enough into the baby mouse, it protected the mice. They survived. That formally proved that NK cells did provide protective immunity. But it also gave us the opportunity for Josh Belke and Joe Sun. They titrated either naive NK cells or NK cells that had previously encountered CMV a month before in a primary host, and asked, what do you need to save the baby mice? You could put in 10,000 memory NK cells that had previously seen the virus. They protected most of the babies. Whereas if you put in the equal number of naive NK cells having the same receptor, in fact, most of the babies died. You do the titration, turns out one of these memory cells is as good as about 10 of these naive cells. And in a plastic dish, we could show that these memory NK cells killed better, and if you stimulated them, they made more gamma interferon on a per cell basis than their naive counterparts. So what we've been doing subsequently is asking, what does it take to generate and maintain these memory NK cells? So a nice system we've used is a competitive adoptive transfer system where we've taken these mice that lack Li49H, and now you can put into them wild-type NK cells, and then genetically marked NK cells that lack your favorite cytokine receptor, signaling molecule, cytokine, etc. Put them in at 1-to-1, infect, and then ask what happens, and are any of these genes needed to make the NK cell memory happen and the memory cells persist. So this summarizes a few years of work where we've done these type of experiments. And the real winner is IL-12 receptor. NK cells need to see IL-12 for you to generate memory. If you just look at the IL-12 receptor knockout mouse, the NK cells are fine. Their repertoire, maturation, numbers are all identical to wild type. But when you actually infect them, they're duds. You get essentially no expansion and no generation of memory. We've done this with a variety of other knockouts. They also need to be able to sense type 1 interferon. If we use an interferon alpha receptor knockout, in fact, you get a blunted response. You get intermediate responses if, for example, you knock out the receptors they need to recognize IL-18 or IL-33. It helps, but it's not essential. One cytokine receptor they don't need is a gamma interferon receptor. In fact, as you know, NK cells make a lot of gamma interferon, but they're making it for their friends. It doesn't need to feed back in an autocrine fashion because the kinetics of the expansion, contraction, and memory generation were identical to wild type. So summarizing, a lot of work we've done in this system, what happens is you have MCMV infection, you get expression of this class 1-like M157 protein. The NK cells then can kill those infected cells through a perforin-dependent mechanism. You then get a proliferative signal, which is driven by IL-12 and type 1 interferon. This process is amplified by co-signals through DAP10 and DNAM. DNAM giving signals through FIN and PKC theta. That leads to the clonal expansion. Typically, like with T cells, once the virus is controlled and you have little M157 around, these cells contract. And this is due to BIM-dependent contraction. We've used NK cells which are BIM knockout. They don't contract, they hang around. Importantly, they in fact, if you hit them again with a second CMV infection, these things can take off and clonally expand again. The second expansion again requires IL-12 and type 1 interferon. For all of the clonal expansion, you do not need IL-15 or any of the IL-2 receptor gamma chain cytokines. However, once the IL-12 has stopped being produced, if you don't have 15 around, the NK cells are dead. They die in about 48 hours without that maintenance factor. So we ask, how specific is this response to CMV? These were studies done by Gundel and Min O in the lab. So what she did was again made some memory NK cells, taking the naive NK cells, expressing the CMV receptor, putting them into the deficient mice, infecting. You now have memory NK cells. A month or two later, you take these out, put them into a second host, and infect again, but this time with either the wild-type CMV or a CMV which has been knocked out for that one gene. What you can see is you get clonal expansion the second time, a robust response, if you have the M157 ligand. If you infect with the virus lacking M157, those memory cells don't go. Okay. However, this virus does replicate nicely, in fact, will kill the mice. So Gundel asked the question of, okay, we've got these memory NK cells that have been exposed to CMV, how would they respond to heterologous other infections? So Gundel set the following experiment. She took again these H-deficient mice, put in some NK cells with the CMV receptor, gave them CMV or left them uninfected. A month later, she then added a few extra naive NK cells that were genetically marked and then infected with either listeria, a bacterial pathogen, or flu. You can now look and see what happens to the memory NK cells that saw CMV versus their naive counterparts in experiencing this new pathogen. And what was pretty clear was, if you looked 4 days after listeria infection, actually the naive cells started taking off and proliferating. The memory CMV cells did not proliferate. Okay. We looked at this directly in the mice by looking at Ki67. This tells you whether the cells have divided or not. And if you took mice which had been previously infected with CMV and looked at Ki67, you can see the memory cells are kind of, ticking over slowly. A couple have proliferated. However, if you give it in listeria infection, the memory NK cells still aren't dividing, hardly at all. However, the naive NK cells in the same animal, when they see listeria, essentially all of them are dividing, saying that memory NK cells are ignoring the bacteria. This was also true if you looked at cytokine production. Now, as you know, listeria induces a quite robust IL-12 response, which can induce gamma interferon. If you looked in those mice, the naive NK cells, in fact, are giving you a nice interferon response. This is 24 hours directly ex vivo out of the infected animal. Memory cells are duds. Very few of them make cytokine to the infection. Basically saw the same pattern with flu infection. So if we now compared the mice which had previously seen CMV, their naive cells versus their memory cells, and infected with flu, again, about 50-60% of the naive cells are now incorporating Ki67. They're dividing. The memory cells, again, are kind of duds. They're not responding. Okay. This was even true in a plastic dish. If we took the memory NK cells and naive cells, put them in a plastic dish with IL-12 and IL-18, 80% of the naive cells, in fact, are making a nice gamma interferon response. And as Shannon told you this morning with human NK cells, he got the same results with the human memory NK cells. They were duds. They don't make cytokine in response to just cytokine stimulation. So we asked the question, why do these memory NK cells respond strongly when they're given the CMV challenge, but respond poorly to cytokines or the heterologous pathogens? And again, as you heard from the story in humans this morning, same thing happens in mice. If you take the memory NK cells and in a plastic dish, give them some IL-12, and then measure phosphorylation of STAT4, which is the downstream signaling molecule which responds to IL-12, the naive cells give you a robust STAT4 phosphorylation. Memory cells are much less efficient. Saw the same type thing with STAT1 phosphorylation with type 1 interferons. The naive cells work better than the memory cells. Okay? So I told you that if you take the memory cells and just give them IL-12, they don't do anything. However, you need to give them an antigen signal. This is a two-signal model. They need antigen and cytokine and gas. So in these experiments, we took these memory NK cells and put them in a plastic dish here with a tumor that we had transfected with the viral ligand. So if you just put in the tumor with no viral ligand, the NK cells don't make any interferon, whether you give them IL-12 or not. However, if you give them ligand for that specific receptor, in fact, that alone will induce some gamma interferon, and you can augment that with IL-12. So they need signal 1, antigen, signal 2, gasoline, IL-12. Okay? So these NK receptors, they're germline encoded. They can't rearrange or undergo somatic mutation like B-cell receptors or T-cell receptors can. But we do see a change. In fact, what we noticed we were looking at some FACS plots in a lab meeting a couple of years ago, is if you look at the cell surface density of Li49H, you can see, you get a bell-shaped curve like you do with any antigen. But when we looked at the memory NK cells that had seen CMV once, there was about 10-fold more receptor on the surface per cell. We actually took these memory cells, put them into the second host, hit them again with CMV, it gets brighter. It's now, a couple of logs higher than on the naive cells. What Yana didn't show you this morning is this exact same thing happens in humans. The Li49C receptor starts out at low levels, but if you look at people who have reactivated CMV, it can be 100-fold higher cell surface density. So I'm calling this avidity maturation, because you now have a much higher density of the receptor. It's going to see lower amounts of antigen. Okay? So summarizing, some of these studies, we saw that with CMV memory cells, they have an increased cell surface Li49 receptor density. They have an enhanced antigen-specific response to secondary infection. They have a robust response the second time, and we've actually serially transferred these cells mouse to mouse through 4 generations, and they keep getting better. They're present in all tissues. There's no special place. It's not like they hang out in the liver or somewhere. You can find them in all tissues. They ignore bystander cytokines. They are looking for CMV. You get synergy between cytokine signaling and Li49H specific activation, and they ignore other bugs that don't have ligands to turn them on. So what you need to make these memory NK cells is you need a good ITAM coupled receptor and a specific ligand. You need co-stimulation, in this case through DNAM1. Stimulating the antigen receptor alone will not drive this response. IL-12 is needed for the proliferation and differentiation of these cells, and you need IL-15 for survival. NK cells are different than T cells in that they cannot make their own autocrine growth factors. So just stimulating, like in a plastic dish with Li49H antibody, will not work. It will make them degranulate, it will make them make cytokine, but they can't proliferate. Somebody else has to make the goodies, like IL-12 or 15, to make them go. So this was all done with transfer, and but we wanted to set up a system where we can track the fate of NK cells in the original mouse. So in the last year, Tsukasa in our lab generated a new tamoxifen-inducible Cree reporter mouse to track the fate of NK cells. So what he's done here is made a construct with an iris Cree ERT. This is responsive to tamoxifen induction. And he knocked this into the NCR1 or NKp46 locus. So to track these NK cells, you can now take these tamoxifen-inducible mice under the NCR1 promoter, cross them with your favorite colored mouse reporter that have a flock-stop flocks with either a yellow fluorescent protein or a red fluorescent protein. And now when you give tamoxifen, that activates the Cree, and now you have red or yellow NK cells. And all their progeny will now be colored forever. This works quite well. You can see that when we did the piloted experiments and Tsukasa gave the mice some tamoxifen, NK cells in this experiment, he got about 30% labeled red NK cells. We played with conditions. We can get this up to 70 or 80%. It's specific. T cells don't get colored. B cells do not get colored. And these are largely myeloid cells, not red. Expression of this fluorescent reporter correlates with when that NCR1 gene is expressed during NK development. So on the most primitive NK precursor, you see no red cells, but in the immature NK cells that now acquire the capacity to express NKp46, now you're starting to get labeling, and you can label all maturation states. Okay. So you heard Hergen give a nice lecture earlier about ILCs. Well, some ILCs, in fact, do express NKp46. And when we do our labeling, in fact, here are some of those ILC3s that are NKp46 positive. They're also red. So you can use this to track some of those ILC populations. So what Tsukasa wanted to do was to now, in the mouse having an infection, track the fate of the cells which are labeled only at the time during infection. So he gave CMV infection and at the same time injected them with tamoxifen. Now what happens is that these NK cells will turn yellow. You will have then NK cells with the CMV receptor, which are yellow, and you'll have some NK cells that don't have the CMV receptor, also yellow, but these NK cells are exposed to that cytokine milieu during the infection. What we saw was, it recapitulated what we saw with our adoptive transfer, in that after infection, you can see that the Li49H positive NK cells, in fact, they clonally expand, contract, and make memory. The yellow Li49H negative NK cells, in fact, don't expand. And you can find them around, but they don't undergo this clonal preferential expansion. However, everyone gets activated. Because, when you give CMV infection, you've got a lot of IL-12, you've got interferons. So if you look at the yellow cells, both Li49H positive and negative, both of them upregulate granzyme B, and both of them upregulate this activation marker KLRG1. The Li49H positive ones, specifically responding to CMV, in fact, do make more granzyme, but both positives and negatives have been activated, increased granzyme, and upregulated the activation marker. I told you previously that you can find the memory NK cells in all compartments. Well, there is one place where now you can compare. You got yellow cells that either have the CMV receptor or not. You can look at their ratios in different tissues. Where they're preferentially expanded is in the salivary gland. That is where CMV stays persistent in mice and humans. I mean, kids get CMV by spitting on each other. That is where you make CMV, and when we make viral stocks, that's where the virus persists, and it persists for the life of the individual. So we think they're there because there's probably some residual low-level replication and low amounts of M157. Okay? One of the reasons they hang around in the salivary gland is that those YFP-positive memory cells, they have higher levels of the integrin, this CD49D that had been previously shown to be needed. That's the way they hang out in that tissue. Okay. Both the Li49H positive and negative NK cells do require IL-12 for their expansion. You get a much more robust expansion of the H positives because they get a antigen-specific signal. But in fact, if you block IL-12 during the infection, in fact, you get fewer memory cells and fewer cells which are just responding to the cytokine. So we wanted to ask, well, all herpes viruses persist forever. Once you're infected, you got them forever. If you got infected in nursery school with CMV, you will have it when you're 100 years old if you're lucky enough to live that long. So we wanted to ask the question, do these memory NK actually need antigen to maintain memory and persist? So Sukaso did the following experiment. He took the reporter mice, infected them with CMV. A month later, he isolates the expanded memory NK cells that are specific for CMV. He isolated the yellow cells which had been activated by cytokine, but not the virus. And he took then some naive NK cells from a genetically marked mouse. This one actually has a red tag on it. Transferred these 3 cells into an uninfected RAG1 deficient animal, and then asked who hangs around. Well, in fact, if you take them out of the absence of a host which has a persistent CMV infection, the memory cells crash. The naive cells work best. They hang around the longest. The cytokine-activated ones hang around okay, but not as well as the naive cell, but the memory cells really are at a disadvantage in a host with no M157. Remember, these memory NK cells with cytokine alone, don't respond very well. So in the absence of antigen, we ask, do these Li49H NK cells, do they need IL-15 to keep going? And we ask the question of, will they compete less efficiently for IL-15 than would naive cells? That's exactly what happens. If you take naive NK cells and memory NK cells, label them with CFSE, and then put them in a plastic dish with IL-15, and then count the number of replication over a few days, you can see that the naive cells proliferate most robustly. They eat the IL-15 better than the memory cells. Memory cells aren't doing well without the antigen hit. So from this, we learned that both cytokine-activated and antigen-specific memory NK cells persist for a month after CMV infection. Actually, we've shown that in a host that has persistent CMV, we can find these memory cells a year later. They can self-renew. Both cytokine-activated and antigen-specific memory NK cells need IL-12 to go. Memory NK cells preferentially localize to the salivary gland. This is the site of latent CMV. Memory NK cells become dependent on antigen-specific signals for persistence. And these memory NK cells poorly compete for survival cytokines when there's no antigen around. Okay? Again, remember, this happens in people too. CMV lasts forever. So I think this allows for the persistence of these memory NK, and if CMV is reactivated, these cells can take off and do a good job. So what about, is this a one-dog trick? Can you get NK memory through other receptors? So there is another receptor which is related to Li49H called Li49D. Mike Bennett and Vinay Kumar many years ago showed that this receptor, it signals exactly the same as Li49H using DAP10 and 12. This Li49D specifically sees H2D of D. It gives you an allo response. So we wanted to ask the question, can you get allo-specific memory? So these Li49D positive NK cells in a black 6 mouse, they will reject H2D splenocytes. Mike Bennett and Vinay showed this 20 years ago now. But what we saw was if we just put in these allo-splenocytes, yep, they're killed and eliminated, we got no clonal expansion and we got no memory. But we've also done the experiment as if we take black 6 mice and give them a tumor transfected with the viral ligand M157, they kill the tumor, but again, there's no clonal expansion and no memory. You need gas. I showed you that the generation of those cells requires IL-12 and type 1 interferon, which are induced by viral infection, but are not induced by allogeneic cells or by tumors. So Sukasa did the following trick. He says, let's provide a viral infection at the same time we give allo. The viral infection will induce the cytokines to give the gasoline, and now the Li49D cells will have the alloantigen to give the antigen signal. So again, what he did was take wild-type NK cells. He put them into a mouse lacking DAP10 and DAP12. He then gave them allogeneic H2D splenocytes, but simultaneously infected them with CMV that didn't have the M157 ligand, which would drive H. And he got memory. You can see that, in fact, if you give the alloantigen without giving the viral infection, the Li49D cells don't do anything. They'll reject the splenocytes, but you got no expansion. However, if you simultaneously gave them a viral infection, you got expansion, contraction, and memory. This again required IL-12. If you do this experiment, this is it's antigen specific. So in that mouse, if you looked at expansion of the various NK subsets, only the NK cells with Li49D expanded and generated memory. The other NK cells in the mouse, in fact, didn't give this clonal expansion, and it requires IL-12. If we neutralized IL-12 with an antibody during this experiment, in fact, we got no allo-specific NK cell memory. These allo-specific Li49D NK cells have a memory phenotype. They've upregulated KLRG1, they've downregulated Li6C, they have kind of low DNAM1, exactly like the Li49H viral story. Okay, so we've got now 2 related receptors where we've demonstrated clonal expansion and memory. How about other NK receptors, like NKG2D? Now this receptor, as you know, is expressed on all NK cells as well as on T cells. And what this receptor does is look for cells that are unhappy. So there's about 10 ligands, endogenous ligands for NKG2D. When cells become unhappy, they upregulate these stress-induced proteins. Li49D will bind, and they'll kill those cells. In mouse, in fact, there are kind of two flavors of this NKG2D receptor. On resting NK cells, the NKG2D receptor signals through DAP10 only. This gives you this PI3 kinase and VUV1 pathway of activation. This is sufficient to trigger killing and a little bit of cytokine production. In mice, but not humans, in fact, activation of mouse NK cells induces the generation of a spliced form of this NKG2D called NKG2D short, which was identified by Colonna's group and Rollet's group, that now can couple to DAP12 and give you the ITAM signal. So we thought, maybe we can get some memory here. So Shikasa did the following experiment. He took mice which are DAP10, DAP12 knockouts, so these mice have no expression of NKG2D at all, or Li49H at all, and he put in a combination of 3 cells. Wild-type NK cells, DAP12 knockout NK cells, and DAP10 knockout NK cells. He then infected with a virus that lacked M157, so you don't drive H responses, and also lacked M152. So many years ago, our lab and Stefan Janik showed that M152 kills expression of NKG2D ligand in CMV-infected cells. So if you take cells and infect them with this virus lacking M152 through 57, you can see some upregulation of RAY1, which you do not see with the wild-type virus, because the wild-type virus kills RAY1. Also, with the wild-type virus, you get M157 expression on the infected cell, but with the knockout, it also lacks 157, so you got no M157 ligand. If he took and put those 3 kinds of NK cells into the mice and then infected with that mutant virus, in fact, the cells just declined and decayed. We had no expansion at all. This said that NKG2D signaling alone is insufficient for expansion and generation of memory NK cells. This is even in the context of a viral infection where you got the IL-12, you got the interferons. Okay. So we thought, well, maybe there's just not enough RAY1 around to make it work well. Well, Stepan has recently made a nice viral mutant in which he took CMV and he knocked into the M152 locus mouse RAY1 gamma gene. Now when you infect cells with this RAY1 gamma CMV, you can see you got tons of RAY1 expressed. So Tsukasa did the same experiment in that he took now wild-type NK versus NKG2D knockout NK cells, put them in this host that doesn't have any of those receptors, and infected with the virus, which is expressing the hell out of RAY1 gamma. In fact, this RAY1 can augment the Li49H response of the Li49H expressing NK cells. So you get a little augmentation. However, we got nothing when we did the same experiment signaling through just NKG2D and not Li49H. So what this told us was NKG2D signal alone is not sufficient for effector NK cell expansion or memory differentiation. However, it can enhance the Li49H positive NK cell response. So as I mentioned to you before, it had been shown that by Colonna and by Rollet that if you take DAP10 knockout NK cells and soak them in IL-2 for 3 days, in fact, you can induce some NKG2D. Here's wild type in green NK cells, cultured for 3 days in IL-2, have nice bright amounts. The background is the double knockout, DAP10-12. They have no NKG2D. That's the same as the control staining. The DAP10, in fact, you do get NKG2D, and this is because you made that isoform that can pair with DAP12. But the problem is that this is very transient and very low level. So if we took the mice, which are DAP10 knockouts, and infected them with CMV, at day 1.5, in fact, you can see in the DAP10 infected mice, you got some NKG2D. However, by day 3, it's gone. So I think you don't have the persistent DAP12 ITAM signal to drive the clonal expansion and the memory. So lack of NKG2D induced memory likely results from the lack of sustained DAP12 signaling due to only transient expression of this NKG2DS isoform that can pair with DAP12. To get NK memory, you need a good ITAM signal, you need co-stimulation, and you need cytokines. With that, I'll stop and thank you. These are the people I mentioned, most of them who were responsible for it. And in fact, I'm looking for a postdoc right now, so if anybody's interested, they can email or talk to me. Thanks very much.
So IL-15 seems to be so important in maintenance of the memory response.
Yes.
Who's providing that?
So from Avro Ma's study, probably almost any type of myeloid cell. So Avro Ma has shown, he was the one who knocked out IL-15 or IL-15 receptor, and they have very few NK cells, but he did a conditional knockout and then looked at NK cell numbers. Macrophages can make it, dendritic cells can make it. It's mostly some type of myeloid cell when he took the flux and crossed it by the various crees.
Okay.
Yeah.
Okay. Great. Are there more questions?
That's really nice, Lewis. So I just have a question regarding the survival of the memory NK cells, because you nicely showed that they needed antigen.
Yeah.
But in humans, quite a few of the memory NK cells seem to not express any DAP12 coupled receptors. So do you think FC receptors could step in and help them compete for survival signals?
Yeah, I mean, you might have the situation of if you have a CMV antibody, right? It could signal through the FC receptor and give you the tickle.
Or even heterologous?
Well, I showed heterologous pathogens don't work. You have to have something, you have to have a good receptor. The FC receptor is good. We know that's the case, right? And it can drive proliferation. So my guess would be in humans, it might be a CD16 signal.
So really very nice story. So how do you think that evolutionary it has been selected, which receptor and maybe which pathogen would induce this type of NK memory? Do you think there is a certain selection for certain pathogens that we need to control better, or?
Yeah, I think we get lessons from that. I think that one of the good explanations for CMV is we co-evolve with CMV, right? And it stays there forever. So I think that's a good rationale. And we know what happens if you're immunosuppressed. The CMV comes back and kills you if you don't have antivirals around. So through evolution, that's telling us the story. We've also learned from people who don't have NK cells, like flu is not a problem for those people. And in fact, if we deplete NK cells and infect with flu, it makes no difference whatsoever. So there are certain pathogens that through evolution, NK cells have paid attention to.
Luis, I suppose memory NK cells generated by CMV infection, some of them co-express Li49D.
Yes.
Are they highly responsive to H2D stimulation?
Yeah. Actually, we've done the experiment in the opposite direction. We made the Li49D memory cells and then took those and tested their responsiveness to CMV. They work better. because the cell has both receptors. I think the internal wiring has made them more efficient so that you can hit them through either of the activating receptors and get a better response.
So it's not just upregulation of the receptor, but something signaling is upregulated in memory. Is that right?
Yeah, I mean, we've obviously done, microarray experiments with memory NK versus naive. There's a bunch of genes that are different, and I don't know so far which ones are most important. There are some survival genes that are preferential, but others I don't know why they're giving a better cytokine response or better killing response.
Lewis, terrific talk. I have a question about the potential of using NK cells for vaccines.
Yeah.
And I mean, given that you showed with the CMV model, you need to continually give the antigen to maintain memory.
Yeah.
It would seem likely that you'd either have to have a vector which is going to trickle the antigen in over a sustained period or boost the vaccinee to maintain memory for a long time. Would that be a reasonable conclusion?
Maybe so, yes. I mean, the only thing I think that may help you out is that we've shown, and Yannen's group has shown, that in humans, the memory NK cells, if you stimulate them through the FC receptor, CD16, that can give them a decent signal. So, yeah, so maybe in humans that takes care of the problem. Mouse ADCC is really lousy, though, compared to human.
So evolutionary speaking also, this controller CMV seems to come at a high price because you would be less able to handle other pathogens, it seems. So is there any clinical impact of that?
Well, there's a lot of studies saying, in fact, in old humans like me, your CD8 response can be 30% CMV, right?
Yes.
So is that a disadvantage? Maybe so. That's maybe why. Kathy Blish actually has done some experiments and suggesting that a responsiveness to HIV is worse after CMV.
Yes.
Because too much of your repertoire is paying attention to that pathogen, not leaving enough room for the other ones.
I'm speculating, of course, of the findings that we have that some vaccines induce negative non-specific effects. So they give you an ability to combat the actual pathogen that you're vaccinating against, but they seem to hamper your ability to handle other unrelated pathogens. Could this be related to some NK cell phenomenon?
Yeah.
Any more questions? I actually have one more. So you just briefly mentioned the self-renewal of these memory NKs.
What's that?
The self-renewal of the memory
Yeah.
cells. How's that regulated? Do you have any idea?
How is what regulated?
How the self-renewal regulated? And they keep
this-
their memory function in the end.
Well, I think, if you have ligand in the host, they turn over. We've done BRDU labeling and Ki67. They turn over slowly, probably as they pass through the salivary gland or something, to keep them self-renewing for as long as the ligand's around. And that's giving the survival signal as long as you have some 15 around also to keep them alive, that combination.
And how long is the lifetime of an NK memory cell?
Well, I think the individual cell itself is about a month, but if it's self-renewing, they can hang around forever, right?
So I wonder whether this is also something happening in the myeloid cells that are trained by innate memory.
Yeah.
Okay.
Thanks.
Thanks a lot. It was an excellent talk and good discussion.